Hello and welcome to part 2, step 6 and 7 of glycolysis. So in this video I will tell you further details about it, okay? So basically I think in my previous episodes I told you to just write down the steps, the words of the substance involved. Like for example here, step 6, I would write glyceraldehyde 3-phosphate, double arrows, to become glycerate 3-phosphate, and then arrow with phosphate group and NAD plus to become 1,3-BPG, for example. So that is the way for your brain to remember things. So what exactly do I mean by that? So after writing on paper like 10 to 15 times, the steps, your brain will start to memorize it better, because that's how it works. That's how the brain works. Exercise makes it to perform better. So here we go. Step number 6 involves turning the glyceraldehyde 3-phosphate into the 1,3-biphosphoglycerate. But first of all, most of the book, I think, or material will not explain to you why it happened this way. I will. Okay. So first of all, you see an aldehyde group here. The enzyme involved in this step 6 is called glyceraldehyde 3-phosphate dehydrogenase. Of course, once again, they use the whole name of the substrate, and then enzyme to dehydrogenase. De mean to remove a hydrogen. Removing a hydrogen is the same as an oxidation, isn't it? Yeah, it's oxidation. So what is oxidation? Is the addition of an oxygen or the removal of a hydrogen. Okay, in this case, you have an aldehyde group, and it will act on that aldehyde group to turn it into a carboxylic acid group, COOH here, carboxylic acid group. So that was an oxidation. Okay, so it turned this glyceraldehyde 3-phosphate into glycerate 3-phosphate. So this is an indication that there's a carboxylic acid group over here. Glycerate 3-phosphate. There you go. Now, there should be a lot of NAD plus, which is the electron transporter, in the environment of the cytosol, and also there should be a lot of phosphate group floating around as well. Free phosphate group, that's what I would say. And they will act over there to remove a H. Let me tell you that you still have that enzyme floating in the environment, okay? So you still have the same enzyme here. You would still have the same enzyme here. So removal of a hydrogen, dehydrogenase, there. That's the action of the enzyme as well. And then removing this one, the phosphate will jump in to the picture to create this one, 1,3-biphosphoglycerate. Where will the hydrogen go? Of course, it will go out to the environment, become H+, and then that H+ will be captured by the NAD. So it will become NADH, electron transporter. I will transport the H somewhere else. I'll tell you later. Now, step 7 involves what? Turning, before I go to step 7, I would like to tell you that this 1,3-BPG is the first high energy intermediate that is formed. Why is it intermediate? Because look at this one, it has 2 phosphate group, and this one is C, the bond OO, is an ester group. Ester, you remember ester? Ester group. So this one, the bonding in between them, this bonding in between is called phosphor-ester linkage, and that is a very high energy bond. When you break it, you release a lot of energy to the surrounding. Okay? That is the reason why this whole thing is a high energy intermediate that is formed. Now, step 7 involved that 1,3-BPG to be turned into 3-phosphoglycerate. So what happened here? Exactly what I told you before, removing of this phosphate group. Okay, so when you put a phosphate group into something, you need a kinase. But when you want to remove that phosphate group, you also need a kinase as well. So here, a kinase enzyme that act on the phosphoglycerate. There you go. And the environment would also need to have magnesium ion as well. And then there would also be a lot of use-up battery, I would call it, ADP, adenosine diphosphate. This is the, how can I say, use-up form of the ATP. So when the ATP is used up, it's going to become ADP. So in the cytosol, there should be a lot of ADP because the cell, obviously, they use a lot of energy as well. So the ADP will be there and accepting this phosphate group with the help of the kinase enzyme to become the ATP. And this is the first ATP generated. Okay. Right. So after this one is removed, this one will become a glycerate. You know, it doesn't have the H anywhere to put in there. So it's still like a negative charge over there. Okay. And mind you that, remember what I told you before? 1 glucose will become 2 GAP by step number 5. So until step number 7, you should have 2 molecules of the 3-PG. And you should also have 2 ATP being produced as well. Okay. I'll see you in my next part.